Hello, everyone, and welcome to today's webinar, Investigating Antimicrobial Mechanisms of Action Using Automated Genome-Wide and Gene-Specific Editing. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Alia Clark-ElSayed, Senior Research Associate at Inscripta. Alia, you may now begin your presentation.
Thank you for the introduction. Today for the webinar, I'm going to be talking about how we used the Onyx genome engineering platform to study the mechanism of action of two different small molecules. To do this, we built genome-wide as well as gene-specific libraries. We then used these libraries in selection experiments with the addition of the small molecules. And then after the selection experiments, we analyzed the distribution of the different edits within the library, and this allowed us to make hypotheses about how we think these small molecules are acting on the cell. Both of the small molecules that we studied in this work come from plants originally, and so I'm going to start off by giving some background on antimicrobials of plant origin. I'm then going to talk about the Onyx platform and how we actually built the genome libraries. I'm then going to talk about the work we did to study the mechanism of action of the first small molecule, p-Coumaric acid, and then I'm going to talk about the work we did to study the second small molecule, Resveratrol. So antimicrobials of plant origin, they are typically used by the plants as defense mechanisms and tend to be secondary metabolites for the plants. They have a relatively low toxicity compared to synthetic antibiotics, and they have a lower incidence of drug resistance than antimicrobials from other sources. Shown here are two plants that I think we're all familiar with. On the left is elderberry, and that was found to have antimicrobial activity against clinically relevant strains of bacteria. And then shown here on the right is dill, and that was shown to also have antimicrobial activity against bacteria due to the terpenoids that it produces. So the antimicrobials of plant origin, for these reasons outlined here, these are very valuable small molecules. And as a result, there could be desire to produce these small molecules. However, there's oftentimes challenges associated with the isolation of the antimicrobials in the plants themselves. This is typically due to the fact that they're produced in very small amounts. And there's also challenges with the isolation because it oftentimes requires many different chemical conversion steps, which could make the processes very difficult and time-consuming. And so as a result, it's possible to port the production pathways over into microbes, which would allow for more scale-up in production. An example of this is artemisinin. This is used as a treatment for malaria. Artemisinin is derived from the plant Artemisia annua. However, shown here is an outline of this production pathway in yeast. So here in the yeast, the acetyl-CoA, through different enzymatic conversions, is eventually converted into artemisinic acid. and then through chemical conversion is converted into artemisinin. And this would be just one example of how you could use microbes to produce such a compound. However, if you don't actually know how the antimicrobial is acting on the microbe, it could make the bioproduction of the small molecule difficult. And so for the work that I'm going to be talking about today, I'm going to talk about a way that you could easily study the mechanism of action of a small molecule. Here I'm going to outline a summary of what we did to study the mechanism of action of our two small molecules. So shown here in a schematic is this small molecule that is acting on your cell in some way, and you don't know what the mechanism is. So with the Onyx, it allows you to design many different types of genome libraries. Here are 3 examples of the genome libraries which we used in this work. So with the Onyx, you can make genome-wide targets, such as all transcription factors, and you can make different insertions or deletions to these genome-wide targets. You're also able to make precise edits to essential genes, such as RPOA. And you're also able to target entire cellular pathways. And in the work that we did, we built a library that targeted specifically the cell division pathway as in addition to an entire morphology and protein folding library. So once you have your libraries designed and built, you're able to then use them in selection experiments. So you would grow your cell library with the addition of your small molecule. And then after you complete your selection experiment with the Onyx, all of the designs are trackable. And so you're able to look at the distribution of the designs within the library. And so when you do that, it would be possible that you would see certain mutations which enrich upon the addition of your small molecule, or certain mutations which deplete in the population. I'm next going to walk through the entire Onyx platform from start to finish for building the libraries as well as doing a selection experiment, and how you would actually go about doing this. So with the Inscripta designer software, this makes it very easy to design all of your genome libraries. And you can make between 100 to 10,000 different designs per library. And this would be the design phase of the platform. Next would be the generate phase, and this is where you're actually building your libraries. So shown here is the Onyx instrument, and here are all of your consumables. And this makes it very easy to just load your consumables into the instrument and push a button, and then the instrument will run, and you'll then end up with a pool of cells which contain your library of designs. Next would be the test phase. So you would be able to perform a selection experiment or a screening experiment. And then with the genotyping kits and the analysis software, you would be able to look at which designs are actually present in your cell. And then with the learn phase, you would use the Inscripta resolver software to enter in all of your results from the genotyping kits and computationally assess your cell library composition. Here I'm going to walk through the E.coli Onyx selection workflow, and this was what we used for our work. This was done in E.coli, so I'm going to outline that. So you will start with your E.coli input cells. These will then be loaded onto the Onyx instrument for an edit competency run. And this will then allow your cells to be editable with the Onyx technology. Once you have your cells that went through the edit competency run, they are then able to go through the Onyx genome engineering run, where you would input the DNA, the design DNA that is specific for your library. And then after this run, you will end up with your pool of cells, which will be edited. After you have this pool, these cells are then able to go through a selection experiment. After the selection, you will then extract the genomic DNA from your cells, and then they will be sent for NGS sequencing, and then that data will be analyzed, and this is what will allow you to make the association between the phenotype and the genotype. The Onyx chemistry is what really enables the scale of having this many edits in your population, and then also to have the trackability of all of your different designs. Shown here on the left is the Onyx chemistry. So you have the guide RNA, the donor DNA, as well as the barcode all connected as one piece. And this barcode allows for the trackability. And we define trackability as the ability to identify the reagents associated with a cell in a complex cell library. With our barcode diversity assay, this is what allows you to actually figure out the design distribution of your cell population. And so when you have your culture here, you'll extract the genomic DNA, and then using that DNA, the barcodes will be PCR'd, and then these amplicons will then be sent for sequencing. So shown here are the two small molecules which we used in this work. On the left is Coumaric acid. Coumaric acid is a cinnamic acid. And then here on the right is Resveratrol, which is a stilbenoid. Coumaric acid has numerous beneficial properties. So it is antioxidant, it has anti-tumor properties, as well as anti-inflammatory properties. It's found in multiple common day foods, including cauliflower, onions, as well as radishes. Resveratrol also has different beneficial properties. It is cardioprotective, anti-inflammatory, and it is derived from coumaric acid. And it's found in grapes, shown here as a wine press, and then also passion fruit. We chose these compounds for our selection experiments because they really represent two different use cases for the Onyx for studying the mechanism of action of antimicrobials. So coumaric acid, we had prior data suggesting that it was affecting a specific cell target, RPOA. And so based on that, we were able to start off by making a more specific library to that due to prior data. And then resveratrol, when we started looking at this in the literature, there were multiple different hypotheses of how resveratrol could be acting. However, we didn't really have any definitive data suggesting the mechanism of action, so we really didn't have a lead target to go off of. And then also because resveratrol is derived from coumaric acid, they serve as very good controls during the experiment for one another. So for the selections that we did with coumaric acid, what we saw after looking at the data from our libraries was that it really showed in RPOA that there were a pocket of enriching mutations and then a pocket of depleting mutations. And these really clustered nicely onto the structure of RPOA, such that there were a lot of enriching mutations in the N-terminal domain and then a lot of depleting mutations in the C-terminal domain. So previous work at Inscripta was done in collaboration with Marcus Ehrgard's lab at DTU, such that an initial genome-wide library was built, and this had 992 barcoded strains. It had 373 adaptive laboratory evolution variants, which were identified in Marcus Ehrgard's lab. It also had 168 insertions of mobile elements, as well as 442 knockouts and N-terminal frame deletions. Once this pool of barcoded strains was built, it was grown in biological triplicate and then grown in M9 with glucose and with the addition of 7.5 mg per ml Coumaric acid. After these cultures were allowed to grow for the duration of the selection, the genomic DNA was extracted and the PCR and the barcodes were PCR'd, and these amplicons were sent for sequencing. Here on the left, we're looking at a graph that it's outlining the enrichment of the different designs, and then here on the right, we're looking at a structure of RNA polymerase with the alpha subunit shown in orange here. So shown here on the left in this graph is the log twofold change of the designs in the Coumaric acid over the M9 abundance. And so anything here on the right would be enriching in the population, and anything shown here on the left would be depleting in the population. And so that these ones that were enriching, we would say that these are more tolerant to the Coumaric acid, whereas the depleting mutations would be less tolerant. And when we actually look at all of these different designs, they are all in RPOA, and when they are mapped onto the structure of RPOA, the enriching variants map very nicely here onto the N-terminal domain, and the depleting mutations shown here in red map onto the C-terminal domain. So this really suggested that there is something going on between RPOA and the Coumaric acid. So a little bit about RPOA. So RPOA is the alpha subunit of RNA polymerase. It's an essential gene. It is a homodimer, and it is really the first step towards the dimerization of RPOA is really the first step towards the assembly of RNA polymerase as a whole. The N-terminal domain has interaction with different transcription factors, and there's also the dimerization domain in the N-terminal domain. The C-terminal domain also has interaction with transcription factors and weak interaction with the DNA. So based on the results from the previous selection experiment, it showed us that there's something going on with RPOA. And so the next library that was built was a amino acid saturation library against RPOA. And in this library, every amino acid was changed to eat every other amino acid as well as a stop codon. And this library generated a pool of 6,880 different variants. This library was grown in biological triplicates, and it was grown in minimal media with glucose and then with ethanol, because ethanol was used to dissolve the Coumaric acid, and then at 3 different concentrations of Coumaric acid, 0.5, 1, and 1.5 mg per ml. These cultures were all allowed to grow for 48 hours, and then similar as before, the genomic DNA was extracted and the barcodes were PCR'd, and then these amplicons were sent for sequencing. The results from this selection experiment were pretty similar to, or they reinforced what we saw in the first selection experiment. So shown here on the x-axis is the amino acid position, and then shown here is the log-fold change of the design in the concentration of the Coumaric acid over the control. And shown here are the different concentrations of Coumaric acid. Here I've outlined the N-terminal domain, the linker, which connects the N-terminal domain and the C-terminal domain, and then the C-terminal domain. What we see here in the N-terminal domain is that there's this little cluster here that's clearly enriching in the population. And then in the C-terminal domain, there is this cluster here, which is depleting, and then also a cluster here, which is enriching. And what you can see as you increase the concentration of Coumaric acid is that the enriching pockets become more enriching, and the depleting pockets definitely become more depleting as you go across. So here I'm going to talk a little bit about where these clusters of mutations actually are on the structure of the RPOA. So as I said before, there are 2 clusters of enriching mutations. The first cluster is at the C-terminal domain, and this is at the site of interaction with transcription factors. The other enriching mutations cluster is at the N-terminal domain, and this is at the dimerization site, and then also at a site distal to the dimerization site, which is at the site of the interaction with transcription factors. The depleting mutations cluster maps onto the C-terminal domain and is at the site of the transcription regulatory surface. So based on the results from the previous selection, this really confirmed to us that there is something going on with having an enriching cluster of mutations and having a depleting cluster. And so the next 2 libraries that we built were libraries that were designed to engineer even greater sensitivity and tolerance to the Coumaric acid. So the way that this was built was that we started by just looking at the enriching score, which is correlated to how enriching or depleting the mutation is within the population. And then we plotted that against the position. And then this data was used to generate these libraries. So the way it worked was that there was a scanning 15 amino acid edit window. And in every edit window, the libraries was designed in a way that there would be 3 to 8 amino acid mutations in the window. And the chance of a mutation appearing in the window was equivalent to how enriching it was in the population or how depleting it was in the population. So if you took the enrichers library, these mutations here, which have the greatest amount of enrichment, you would expect those to be the most greatly represented in the library. And then the opposite for the depleters library, such that these mutations here, which had the most depletion, these would be the most overly represented within the depleting mutations library. And so once we had these libraries built, we used these for a selection experiment where we increased the concentration of Coumaric acid. So similar as before, we took the libraries and they were grown in biological triplicate. and then they were grown in minimal media with glucose, and then plus the addition of DMSO. And in the previous selection, ethanol was used as the control. However, in this experiment, we used DMSO because we used DMSO to dissolve the Coumaric acid, and this was because we were using higher concentrations, and we saw that when we used ethanol that it was just too much ethanol for the cells, and that was causing them to not grow. So instead, we switched to DMSO. And then we had 3 different concentrations of Coumaric acid, 1, 2, and 3 mg per mil. So then the same as before, these cultures were allowed to grow for 2 days, and then the DNA was extracted and the amplicons were sent for sequencing. Shown here on the left is the results from the enriching mutations library, and then shown on the right is the results from the depleting mutations library. I again put in the sections which correspond to the N-terminal domain, the linker, and the C-terminal domain. Shown here on the y-axis, we're looking at the log 2-fold change of the highest concentration of Coumaric acid, 3 mg per mil, over the DMSO control. And similar as before, we definitely see these pockets here in the N-terminal domain, which are enriching in the enriching mutations library. And then there's also this pocket here that we saw before, which is enriching in the C-terminal domain. And then in both libraries, we also saw enrichment within the linker section. And in the depleting mutations library, the results aren't quite as obvious as the enriching mutations library, but however, here in the C-terminal domain, you can definitely see that there is this pocket here, which is showing a lot of depletion. Here I'm going to outline the hypotheses of what we think might actually be going on with the Coumaric acid and in these different pockets of mutations. So we think that the enriching mutations might allow for some sort of an increased transcription factor stress response. And this is inferred because the mutations are in the area that binds to the transcription factors. It's possible that the depleting mutations might be doing the opposite, so hindering the stress response by disrupting the transcription factor binding. And then it's possible that the Coumaric acid could be actually affecting the dimerization site to prevent the dimerization of RPOA. So similar to transcription, another necessary cell process is cell division. So for the work we did to study the mechanism of action of resveratrol, based on the results that we saw, we think that the resveratrol is having some sort of disruption to the cell division machinery assembly in the cell. There are a couple of different proposed mechanisms of action of resveratrol in E.coli. One proposed mechanism of action is that it could be producing reactive oxygen species, which is causing damage to the DNA. Another proposed mechanism of action is that it could be suppressing Fit-Z, which is essential for the formation of the Z ring during cell division. Because there are a couple of different hypotheses for what could be going on, our first library that we wanted to build, we thought should kind of be a more general library rather than with the Coumaric acid where we built a gene-specific library. With this, we built a genome-wide library that was kind of designed to target a lot of different genes, specifically transcription factors, and see what the results of that suggest, and then based on that, kind of narrow it down to what we think could be going on. So with our transcription factor library, we designed it in a way such that we took every transcription factor, and in front of it, we inserted two different promoter insertion mutations. These were both medium strength promoters inserted at the -16 nucleotide position. And then we also made two knockout mutations so that a triple stop codon was inserted at the 10th and the 15th amino acid positions. We took this transcription factor library that had these 4 different designs against every transcription factor. We grew it in biological triplicate, and these triplicates were then grown in minimal media with glucose, DMSO, which was used to dissolve the resveratrol, and then 114 micrograms per mil resveratrol and 228 micrograms per mil resveratrol. These cultures were then allowed to grow for 48 hours, and then same as in the Coumaric acid selection, we took the DNA and PCR'd the barcodes for the designs, and then we sent these amplicons for sequencing. So the results that we saw from this were that resveratrol is having a really large impact on 2 specific transcription factors outlined here that we thought were interesting. The first one is SOX-S, and the second one is DICA. So SOX-S is an ROS response regulator, specifically an activator. Here we're looking at the log 2-fold change of the design counts in the 228 micrograms per mil resveratrol, which is the higher concentration, over the DMSO control. And here we can see one of the promoter insertion mutations, which is the most enriched in the population. And then here is the second SOX-S promoter insertion. Here we're looking at a similar representation of the data. Here we're looking at the enrichment score and then the promoter strength across the x-axis. And we can see that with SOX-S, as we increase the promoter strength, that the enrichment score increases. The second transcription factor that we thought had an interesting result was DICA. DICA is a cell division regulator, a repressor, and it also had a similar result with the increase in promoter strength. So shown at 0, there's very little enrichment of this design. And then as the promoter strength is increased, there is a greater enrichment, and this does taper off here for the second promoter strength. So DICA represses the operon containing DICB, which interacts with min-C to inhibit Fit-C ring formation. And this second result here, this was what kind of suggested to us that there could be something going on between resveratrol and the cell division machinery. And we used the results from this library selection to kind of inform the next library that we'd build. The next library that we built is what we're calling the Morphology and Protein Folding Library. This library has a knockout, a medium, and a high-strength promoter insertion in front of all genes encoding proteins in these pathways. And so once we had this library built, the same as before, we grew these in biological triplicates, and they were then grown in minimal media with glucose with the addition of DMSO, because that was used to dissolve the resveratrol, and then 114 and 228 micrograms per mil resveratrol. These then grew for 2 days, and then the genomic DNA was extracted and the barcodes were PCR'd, and then these amplicons were sent for sequencing. What we saw in this selection was that the most enriching mutations involved changes to the expression of different cell division proteins. So the first cell division protein that had a lot of enrichment was Fitz-A. Fitz-A colocalizes with Fitz-C to the septal ring structure. Fitz-L, which is also an essential cell division protein, and it's required for the localization of Fitz-I. And lastly, Tol-Q. Tol-Q is involved in peptidoglycan processing during cell division, and it's recruited to the divisome at the same time as Fitz-A. Here, we're looking at the log two-fold change in the design counts of the highest concentration of resveratrol over the DMSO control. And here I've outlined in different colors these different designs changing the expression of these three different proteins. Shown here is the enrichment score on the y-axis, again with the promoter strength on the x-axis. So here for Fitz-A and Fitz-L, you can see that there is very little or no enrichment with the knockout. However, as you increase the promoter strength from the medium strength promoter up to the high strength promoter, there is definitely a drastic increase in enrichment. And then for Tol-Q, we actually see the opposite. So there's enrichment, a decent amount of enrichment in the Tol-Q knockout. And then as the promoter strength is increased, the enrichment drastically drops off in the population. And shown here again is the order in which these proteins are recruited to the cell division machinery. And so based on this data here, we're not able to make any definitive claims about how resveratrol is acting and like what its specific target is in the cell. However, this does suggest that resveratrol is having some sort of impact on the cell division machinery, just due to the fact that changes to the expression of these three cell division proteins have such drastic changes in enrichment when they're grown in the presence of resveratrol. And finally, I just want to make a few conclusions about how we use the Onyx genome engineering platform to study the mechanism of action of these small molecules. So the genome engineering platform enables the design and the build of the libraries to be very simple as well as very quick. So for this work, we built a total of 5 genome libraries. This had a total of 23,977 total variants. For each of the genome libraries, the hands-on time actually spent building them for each of the libraries would be around an hour, if not a little less. The trackability of the designs in all of these libraries allowed us to have such a high throughput selection experiment. Using the barcode diversity assay, we were able to look at the enriching and depleting designs in the population and analyze the distribution of these designs. And then finally, the Onyx workflow really is ideal for studying the mechanism of small molecules because it allowed us to identify for Coumaric acid a specific functional region of a single protein, RPOA, which is being affected by the Coumaric acid. And then for resveratrol, it allowed us to identify a specific functional class of protein which is being disrupted by the resveratrol. And with that, I would like to thank you for your time for listening for today's webinar, and I'd be happy to take any questions now.
Thank you, Alia, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click in the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, why did you decide to study Coumaric acid and resveratrol?
So we decided to study these two small molecules for a couple of different reasons. The first is that these are really interesting small molecules due to the different properties that I outlined in the presentation, such as the fact that they have anti-inflammatory properties. And then another reason is that they represent two use cases for the Onyx for studying small molecules. So with Coumaric acid, we had prior data to go off of to have some sort of a lead to design a more targeted genome library. Whereas for resveratrol, we didn't really have a whole lot to go off of. And so because of that, we designed a more broad genome-wide library targeting all of the different transcription factors. And these also served as really good controls for one another because resveratrol is derived from Coumaric acid.
Great, thank you. Next question. Are there plans to build more targeted libraries to study the mechanism of action of resveratrol?
Yes, so for the next library that we're planning to build to study the mechanism of action for resveratrol, we're going to build different saturation mutagenesis libraries targeting the proteins identified as enrichers and depleters in that selection experiment. So that would be the FITS-A, FITS-L, TOLQ, and DICA. And then once we have that library built, we'll be using that in a subsequent resveratrol selection experiment.
Thank you. Next question. Did you test the impact of Coumaric acid and resveratrol on other genes?
Yes, so we studied what was going on with Coumaric acid. We used that in a selection experiment with another saturation mutagenesis library designed against an essential gene, specifically FITC. And we did this just to see if there would be a similar response as what we saw with the RPOA saturation mutagenesis library. And we did not see any sort of clustering of enriching and depleting regions. And then we also studied resveratrol against the RPOA saturation mutagenesis library to see if that would also have the same effect, and we did not see any of the clustering, enriching, or depleting regions.
Okay. Next question. Maybe I missed hearing this, but can this Onyx system also be applied to yeast and fungi?
Yes, so you can use the Onyx genome engineering platform to design libraries in yeast, specifically S. cerevisiae. And in yeast, you can have between 100 and 6,000 different unique variants in a library.
All right, thank you. Next question. Great presentation. Why does resveratrol being derived from Coumaric acid result in it serving as a good control? Also, would you recommend the Onyx system for studies of larger molecules?
So we were thinking that you're able to use the Coumaric acid and resveratrol as controls for one another, just because they are of like a similar size and relatively like similar chemical composition. And so our thinking was that if we used one for the other and saw the same effect, that might suggest that the result that we're seeing isn't actually due to the interaction between the Coumaric acid and the RPOA. And so we just did that kind of as a control for that.
All right. Next question, how are barcodes added and analyzed, and are they integrated into the chromosome?
So the barcodes are not integrated into the genome. They are on a plasmid, and each of the barcodes is associated with a unique design in your library. And so after you have your pool of edited cells, you're able to PCR amplify across the barcode and then submit that amplicon for sequencing. And then using our Resolver software, you're able to analyze the distribution of each of those barcodes in relation to the entire pool of edited cells.
All right, thank you. Next question. Have you been able to translate conclusions about the mode of action to successful biosynthesis of these molecules in microbes?
So we haven't taken it as far as to kind of like relate the results that we saw here to the actual biosynthesis of these two small molecules. However, I do think that would be very interesting to see if there would be a correlation between these enriching mutations that we saw and having improved successful biosynthesis of the small molecules.
All right, thank you. Next question, would it be possible to study a complex system such as probiotic fermentates?
I think that would be possible to study, just given the wide range of different genome libraries that you'd be able to study or build, I mean. So you'd be able to make a lot of different genome-wide targets. You'd also, I'm just thinking if you have like a specific fermentation pathway, that you could also make different promoter insertions or changes to different expression conditions of each of those genes, encoding those things in your pathway. And I think that could definitely be a good use case for these libraries.
All right, thank you. Next question, what organisms does Onyx support?
So the Onyx supports E. coli and yeast, specifically Cerevisiae.
All right. And we will wrap up with that last question. Thank you again, Alia, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.